Sir, like, will it be okay, like, if I just मैं वीडियो बंद करके like बोलूँ तो चलेगा
ठीक
क्या? Like,
ठीक है। अच्छा,
थोड़ा
इंटरनेट
इंटरनेट का
स्लो।
नहीं तो
हाँ,
इश्यू
हाँ,
होगा.
हाँ, हाँ, चलेगा ना, चलेगा। यस, यस,
Thank you, sir.
यस। नाउ डैश इज योर्स, है ना? नाउ यू कैन स्टार्ट।
Thank you, sir. So, good evening, everyone. I'm Maitri Mishra. I'm a PhD student at the Antimicrobial Research Lab, Department of Biotechnology, University of Mumbai, from the Kalina campus. So, my topic was on biotechnology, especially like my expertise was on nanotech. And in nanotechnology, my expertise was on silver nanoparticles and their different properties. So today I'm going to take just a small part of the actual nanotechnology, which is silver nanoparticles, synthesis and characterization. So firstly, what is the term nanotechnology? So you must have come across the term nano during recent times. In fact, these days nano is like being told about almost everywhere. Like you talk about nano in solar cells, you talk about nano in even sunscreens, you talk about nano in every place. So what is nano? As you know, the best things come in small packages, and also we know that everything, in everything there is matter, like our laptops, our phones, the chair we are sitting on, the pencil we are writing with, the spectacles that we are having, our headphones, everything around us is made up of matter, which is made up of atoms. So nanotechnology is the study of these atoms, the molecules and their interactions. And nanotech is being used in various applications, right from sunscreens to solar cells to medicines to everything. Even our LED screens, LCD screens, they have a little bit of nanotech in them. So nanotech is an interdisciplinary science. It involves physics, it involves chemistry, it involves biology, and a little bit of engineering when you have to play with all these three subjects. So yes, it becomes a very interesting field. It is being considered the next big revolution. It has already brought about a revolution in various things. Like, we have different kinds of solar cells, we have transparent solar cells, we have medicines which can, which have small cameras hidden in them, which allows the doctors to see where the medicines are going and how they are acting on the patient system. We have so many revolutionary things already started because of nanotechnology, and yes, it is going to bring about many more advantageous changes to our um, research. Now, how small is nano? A nanometer is a billionth part of a meter. In the simplest sense, 1 nanometer or nm is 10 raised to minus 9 meter. So nano is for 9. Like 1 nanometer is 10 raised to minus 9 meter. Simple to remember. So nanotechnology involves nanoparticles, and nanoparticles are ranging from 1 to 100 nanometer in length. Any one dimension of these nanoparticles has to be between 1 to 100 nanometer in length. A nanoparticle, like one single nanoparticle, can contain anywhere between 40 to 400 atoms. If it has less than 40 atoms, it is considered a quantum dot. If it has more than 400 atoms, it is considered a microparticle or a submicron particle. So, why should we study nanoparticles in the first place? Now, Richard Feynman in 1959, he had a very famous speech in which he said that there is plenty of room at the bottom. At that time, he was the first person to indicate that there is something called nanotechnology which can be explored, and that we can start considering about atoms and molecules at a nano level and not just at the micro and the bulk level that we normally keep considering them for. The properties of nanoparticles become very different from the bulk matter. Why does that happen so? That is because, as we saw here, the nanoparticles, like one single nanoparticle, contains between 40 to 400 atoms. And normally, as we know, a bulk matter contains like millions and millions of atoms. So, that the properties of those millions of atoms versus the properties of those 40 to 400 atoms. Even if they are the same matter, they become vastly different. And the most unique property amongst that is their high surface area to volume ratio, also called the SAV ratio. Now, what does that mean? It means that the surface area, or like suppose if you consider it to be a circle, like a nanoparticle to be a circle, a nanoparticle is so small that the volume of the sphere of a nanoparticle has very less volume, whereas the surface area will have more surface area. Like, suppose the volume will be like 10 cubic meter, but the surface area will be like 40 square meters or something. Like, just a rough explanation. So this high SAV ratio, what does that do? It makes them highly reactive and efficient in very, very small quantities. So this thing, this highly reactive nature, which makes them efficient in small quantities, is the exact reason why people pursue nanoparticles in the first place, or studies of nanoparticles in the first place. So nanoparticles are of different types. There are many different types of particles, but there are major five different types of nanoparticles. So the first one over here are metallic nanoparticles, which is very simply nanoparticles made up of metals. Simply told, gold, silver, zinc oxide, selenium, they are all metal nanoparticles. Then there come to be carbon based nanoparticles. What are carbon based nanoparticles? They can be the fullerenes, they can be carbon nanotubes, they can be graphene, they can be quantum dots. And then comes dendrimers. Now, what are dendrimers? Dendrimers are star shaped molecules synthesized with tree like structure. They look like a snowflake. Like a dendrimer can almost look like a snowflake. If you have seen how snowflakes look, they are very decorative when you see them under the microscope. So, dendrimers look somewhat like them. They have a compact molecular structure, and they can accommodate various functional groups. And that is why dendrimers are very much used for drug delivery, because firstly they have a very compact structure, secondly they can accommodate various different functional groups. Then comes lipid nanoparticles. Now lipid nanoparticles, as the name suggests, nanoparticles made up of lipids. But the thing about lipids, these lipids, is that they are ionizable lipids. Ionizable in the sense that you can make them positively charged, you can make them negatively charged, and they form a protective shell around the cargo that they are supposed to carry. So if you make them positively charged and you put some medicine or you put some drug in them, so you can direct them to go towards a certain negatively charged or an oppositely charged molecule inside the system of the patient. And then the nanoparticle will disperse there and the medicine will be directly delivered at that place. So, yes, even lipid nanoparticles are very much used for drug delivery in human systems and normal animal systems also. Lastly, there are polymeric nanoparticles, which are solid colloids of natural or synthetic polymers. For example, chitosan nanoparticles, gelatin nanoparticles, alginate nanoparticles, and polyethylene glycol nanoparticles. So, these are the five major types of nanoparticles that exist in nature. There are many more, but these are the five major ones. The applications of nanoparticles, as we have already been talking about, nanoparticles are being used in many different places. The most primary ones being biomedical, drug delivery. They are used in agriculture and food. In cosmetics, they are used in textile. In textile, they are helping to create self cleaning cloth. Like, even if ink or wine or any other stains, like some stains come on your cloth, but the cloth will clean itself. The nanotechnology is helping in that. Then it helps in optical engineering, electronics, because of the high SAV ratio. It helps in metallurgy and metals. It helps in defense and security. Why? Because It helps to create very, very small microchips or solar chips, or, you know, some chips that can help to transfer information from one place to another. And that helps in defense and security. And the same way it helps in energy storage also, like very, very small microchips, such as the small, this are numbers like SIM cards, the small micro SIM or nano SIM that we have these days. It's somewhat like that. Then these nanosystems, yes, in medicine, they are helping in gene therapy, they are helping in drug delivery, they are helping in cancer therapy. Like we told, ki like we can ask them to be very directed, very specific, and they will go to the specific place and disperse, and the medicine will directly go exactly at the target and deliver the drug, which will be very useful. So there is no side effect of the drug because of going to some other in unrequired place in the body, and in a very small concentration it will be very effective. So it helps in the development of proper drugs and delivery systems. It is helping in genetic disorders and ocular diseases. The eye drops that we have these days, they have some lipid nanoparticles in them by the same, which work by the same mechanism. Then it helps in cardiovascular diseases and brain and CNS biomarkers disorders. So now silver nanoparticles or AGNPs in general. Now silver is a transition D block metal. Its symbol is Ag and its atomic number is 47. It is ductile, malleable, shiny, and it has face centered cubic crystals, which look like this. The image that you can see on the right, face centered cubic means looking like this. So, if this is a face of the cube, there will be one atom at the center and four atoms on each of the vertices of the cube. So, this is called a face centered cubic crystal. It is one of the oldest metals known to man, one of the most attractive metals known to man apart from gold. It has the highest electrical and thermal conductivity and reflectivity. And but still, obviously, for obvious reasons, it is not used in electrical conductivity. Copper is used, or tungsten was used before. It has very high antimicrobial properties. In ancient times, royal families were known to carry silver utensils. As we know, like even in our homes, we must be having, chandi ke bartan, chandi ka glass, or something which we take out even during proper, like, festivals ke time we take it out. So that, what would that help? Like, it would kill any microbes which would be there in your food, and that would ensure that you are having all, matlab, no microbes you are having proper food. But too much of that would cause an argyria condition in those royal people, which made them bluish gray. In their skin would become bluish gray, and hence they would be called blue-blooded people. So the unique properties of silver and the unique properties of nanoparticles that we discussed above makes AGNPs in particular very, very useful for various applications. The applications of AGNPs, primarily they are biomedical. They are used for sensors also. Sensors as in they will detect the presence of various metals and chemicals in your sample. They can be used in food, like food packaging, like a packaging of AGNP coated plastic can help in keeping the food sterile for a longer period of time. In agriculture, it helps in better delivery of pesticides and herbicides at a smaller concentration. In healthcare, again, the same thing. Environment, again, as we discussed for agriculture. For renewable energy, it is being used for solar cells and for making more efficient solar cells. And for water treatment, it is being used to kill the microbes in the water and to remove the heavy metals from the water, making it potable. Now, synthesis of AGNPs is mainly by two major approaches. One is a bottom to top approach and one is a top to bottom approach. What is bottom to top approach? Here, bottom means atoms and molecules. They will combine to form certain nuclei, These nuclei will combine to form nanoparticles. So this is a bottom to top approach. Whereas a top to bottom approach is where you have bulk matter like bulk silver, and you break them into fine, fine particles so that you get nanoparticles at the end. This is a top to bottom approach. Now, the bottom to top and top to bottom approach can be considered in three major ways: physical, chemical, and biological, out of which we are first going to consider physical methods. Now, physical methods, the most famous type is laser ablation. As you can see here, what is happening is that you have a metal plate over here, and this metal plate is kept in a liquid which is not reactive to that metal, obviously. Then you stir the metal, you stir the liquid, and it is kept heating at a certain temperature. Now, what happens is here you have a laser beam, and there is a lens to condense this laser beam. So, what happens is this laser beam will hit this metal particle, it will hit the electrons in the metals, so that surface material, the electrons and the atoms in that on that surface where the laser hits the metal plate, it forms a little bit of plasma plume. What's a plasma plume? It's a small sort of bubble which will form somewhere here, and it gets released into this surrounding liquid, which is this greenish colored thing. Once it gets released into the liquid, it will burst because of surface tension, and the bursting of the bubble causes the release of nanoparticles into this fluid. And this is how you get nanoparticles from this laser ablation method. So it first vaporizes and then rapidly cools. So here, this laser beam is causing vaporization, and the liquid is helping in rapid cooling of the nanoparticles which have been synthesized. The next method is physical vapor deposition. So here we were hitting the metal plate with a laser beam. Here we are evaporating the solid into a vapor phase directly, and then we condense the vapor onto a substrate. So here there are three major steps. What are the three major steps? This is a substrate, this is our substrate, and this is the sputtering target. Like from here, you wish to get the nanoparticles from this metal plate. And here, from here, you are getting the sputtering gas inside and it is coming outside also, and you maintain a certain pressure. Now, this sputtering gas is usually argon gas. What does that argon gas do? It hits this metal from time to time. When it hits it from time to time at a certain speed and a certain pressure, these sputtered this sputtered target releases some atoms which then deposits over this thin film. So from a solid state, it becomes a vapor where you can just see some colloids in the air. Like you see some dust particles in air sometimes, वो वैसे दिखते हैं. And then they settle on this thin film. And when you remove this thin film, like when you just close the system and you remove this thin film, you will see that you have formed nanoparticles on this thin film. More methods of physical synthesis include electrical irradiation, gamma irradiation, where we use electricity and gamma rays, evaporation condensation, which is similar to this physical vapor deposition method, sedimentation process, where we try to sediment the uh, particles which have been created. Then there is a ball mill process, which is basically crushing of the metal plate such that nanoparticles are formed. Then there is magnetic sputtering, which is very much used for forming iron nanoparticles for their magnetic properties. Then there is inert gas condensation, and then there are more mechanical methods which involve different kinds of crushing and ball milling methods. Now, advantages of physical methods are that they are very pure nanoparticles. Like because it's only the metal which is involved. Like suppose if it was silver, So only the silver will be there in the nanoparticles. There will be no contamination in the nanoparticles. No use of chemical reagent. The synthesis rate is really rapid. It will form as rapidly as you want it to. And the nanoparticles are usually of a very uniform shape and size simply because the process is very much uniform. It's very much mechanical, so the size and shape is very uniform. But the disadvantage is that the yield is really low. It's like barely 5 to 10%. The control of nanoparticle size is very less. You just can't, like, if you say that you want 40 nanometer size, but the process is such that you get 20 nanometer size. So all the nanoparticles will be 20 nanometer size and spherical in shape. But you wanted 40 nanometer, so that is something which is not possible in physical method. Then it needs sophisticated instruments, as we already saw. You need a laser beam, you need a sputtering target, you need so many things, which obviously require certain infrastructure and certain cost of the equipment, which is not always possible in all the places. A very high temperature is needed again, which needs some special facilities, not possible in all the places. High energy consumption. So, yes, it very much depends on the presence of electrical energy in our the place wherever you are doing this reaction. And it needs a large amount of space because these machines that we talked about, they are actually very large. So, apart from the machine, the cooling system and the jackets of these machines are really large, so they need a very large space in total. So, yes, this was about physical methods. Now, chemical methods are generally these. So, chemical methods can be what? Chemical methods are very simple in the sense that you will have a substrate, you will have reagents, then you will have products, and then you will have some proper system, like a certain some chemical condition in between, like heating or cooling or something like that. So chemical reduction is mainly that. Then there are microemulsion techniques, UV initiated, photo induced, which includes light, electrochemical synthesis, which involves the use of electrochemicals like electrochemical cell. Then there are irradiation methods. There is microwave assisted synthesis where we use microwaves like microwave valley waves. Then there are polymers and polysaccharides, which are used to reduce the substrate into nanoparticles. So, chemical reduction usually involves the substrate containing silver, as we see here, a reducing agent. Suppose this, that is sodium borohydride, NaBH4. Then it causes instant nucleation of these Ag plus ions. So Ag plus ions, suppose there is AgNO3, silver nitrate. Silver nitrate will have silver ions in the solution. They get reduced. Reduced as in um, Ag plus will become Ag0, and this Ag0 nuclei will form on being reduced with sodium borohydride, which will coalesce, or in simpler terms, combine to form clusters of Ag. Now, these Ag clusters will immediately be capped by something called a capping or a stabilizing agent, which in this case is citrate or trisodium citrate. These newly synthesized nanoparticles will rearrange themselves. Like here, the clusters are just like literally that, just clusters, like cluster of grapes or something. But then they just rearrange themselves to form a proper sphere. And then these nanoparticles will undergo further reduction by the citrate molecules, and they will just form a better structure. They will fine tune themselves into a proper structure such that like it goes and undergoes stages of nucleation to finally form this big silver nanoparticle stabilized by citrate molecules. And this will be normally a size of 20 to 50 nanometer. So these will be like very small, which undergo so many stages of condensation and nucleation to form a 20 to 50 nanometer sized silver nanoparticle. In UV or photo induced reduction, if it involves a UV light or the sun as a source of energy. So here we were having sodium borohydride and we were having trisodium citrate. Here we simply have sodium borohydride and a light source. So the light source helps in the reduction of AgNO3, and we see the change in color from yellow to blue, which indicates that some certain nanoparticles have formed. In electrochemical synthesis, we have a proper electrochemical cell where the anode and the cathode are both made up of silver. The anode is a sacrificial anode. Why? Because this anode will be used, like the ions from this anode will be used to form Ag nanoparticles which will settle on the cathode. And the solvent is usually something which is suitable to the formation of silver nanoparticles. A nice acidic solution. Advantages and disadvantages. The chemical synthesis methods gives a uniform shape and size, again like the physical synthesis method. The synthesis rate is very rapid. The methods are very easy to follow. They are not as hard as the physical methods to bring the laser ablation gun or to bring some sophisticated instruments. No, you just have some reagents. It is possible for you to make silver nanoparticles. The yield is very high. If you fine tune them properly enough, the yield can be as high as 70 to 80%. Disadvantages are that the chemicals are very harmful. They are not friendly for the environment. They are not friendly for ourselves also. A little bit of mishandling and you can, just harm your fingers and yourself if you don't take care of proper instructions when forming the reaction. And next is that it has certain impurities and byproducts, obviously, because, like, suppose here there is sodium borohydride and here there is citrate. So, the solution which contains these silver nanoparticles will still have some citrate and some sodium borohydride, which will here be considered as impurities and byproducts. Now, lastly, there is biological methods, which is a green synthesis method. Now, biological methods will involve a biological system. What is a biological system? There are basically four biological systems: bacteria, fungi, algae, and plants. So, any of these systems, any of these cells can be used to synthesize nanoparticles, and the synthesis can be intracellular or extracellular. So, we will just see how that happens. In intracellular, as the name suggests, the silver nanoparticles will be formed inside the cell, and in extracellular they will be formed outside the cell. So suppose like this is just a diagram which is showing this is a bacteria which was treated with these this is a gold nanoparticle example, but silver nanoparticles is also the same. So you treat it with a gold acid with some water. You give it some dark conditions, you heat it and you rotate it for some time for 12 hours. And then what happens is that these bacteria take up this acid and they form gold nanoparticles. But the nanoparticles are inside the cell. So you'll have to break open the cell and you will have to retrieve the nanoparticles which have formed. So this includes some certain downstream processing. Cut to extracellular synthesis, which is very simple. Here, what they are doing is they just have some extracts. The extracts can be anything. They can be your microbial extract. They can be waste extract. They can be fungal extract. They can be plant peels. They can be fruit peels. They can be anything. They can be seeds also, like whatever. You can make an extract out of anything and combine them with a metal precursor and give them certain conditions like stirring is required, mixing is definitely required, and a certain balance of pH and temperature is also required. So this thing will result in the formation of colloidal nanoparticles, and you will see a clear color change from green and yellow to proper wine red color formation. Now, the advantages of biological methods: no toxic chemicals, very easy to scale up. You don't need any sophisticated instruments, you don't need any harmful chemicals. It's very easy to perform. It's cost friendly, it's user friendly. There is no need for any extreme condition. The nanoparticles found by biological methods are known to be very biocompatible, and it's easy to fine tune for different structures. Disadvantages are that the control of size and shape is challenging, but yes, if you fine tune it enough, if you standardize it enough, you can control that. The yield may be low if the protocol is not standardized, but again, if you standardize it properly enough, it can be controlled. The identification of actual reducing and capping agent. Yes, that is the biggest challenge in biological method, to determine what was the actual reducing or capping agent in the entire biological system that caused the formation of silver nanoparticles. And for this, we need to undergo something called characterization. Now, why should we characterize AGNPs? We have to characterize AGNPs to understand their structural properties, such as size, shape, surface charge, etc. Surface properties affect the functional properties. The functional properties will affect the application of the silver nanoparticle. The better the surface property, the better will be the functional property. And the better you know the surface properties, the better you will be able to predict the functional properties of the AGNPs. And accordingly, you will be able to modulate them for their applications. So, AGNP characterization is important to understand the composition, the concentration of silver, the size, the shape, and the surface charge, because all of these will affect the application related properties such as efficacy and biocompatibility, etc. So, how do we characterize them? Firstly, we need to confirm the reaction whether AGNPs have formed also. So, the first thing that you always do is to do UV vis spectroscopy. An absorbance spectrum will confirm whether AGNP formed or no. Then you do transmission electron microscopy or scanning also is possible to understand the size and shape of the AGNP. Then you do selected area electron diffraction or XRD, that's X-ray diffraction studies, to understand the crystalline nature of AGNPs. Then you do dynamic light scattering and zeta potential studies to understand the hydrodynamic properties and the surface charge of the AGNPs. This is done usually on the same machine. And lastly, you do Fourier transform IR spectroscopy. What does that say? It gives you a tentative idea. It gives you an indication of the possible reducing and capping agent of a biologically synthesized AGNP. So going through them one by one, UV vis spectroscopy. So this is how we know UV vis spectroscopy looks. You have a simple light source, you have an entrance slit. This is the basic one, where this is the entrance slit. You put a quartz prism here, it will disperse at a certain wavelength. The exit slit will allow only a certain wavelength to pass through the sample, which will get detected. And over here, the same thing is converted into a signal by the computer, which can be shown as an output to us about the absorbance spectrum or the OD or the transmission of the sample. So, silver nanoparticles are known to have a characteristic absorbance peak at 390 to 450 nanometer with a peak at 420 nanometer. It helps to confirm the AGNP synthesis. It is useful for protocol standardization. Like we will see how it happens. Time, temperature, pH, etc. You can standardize. It helps to understand the stability of AGNPs over time, and it helps to give a possible size and shape also. So we will just see how that happens. So see, these are some figures which have AGNPs being synthesized. And just as see, like in the top left one, what is happening is here this person is trying to standardize the protocol for the best time of the reaction. So just see, like the purple one is telling ki after 10 minutes only this much is formed. Then after 20 minutes a little more. Then after 30 a little more, so on and At 60 minutes, you have the maximum formation of the silver nanoparticles, such that the peak is also very sharp and it's giving you a very nice OD reading at 1.4. And it's giving you exactly at 420, 425 nanometer, which also tells you that the silver nanoparticles have formed. So this tells you that it it can be used for adjusting the time, temperature, pH, and many such parameters of your reaction. Then over here, this is telling what the same thing is happening, but here they are doing it for pH. So just say pH 11 gave me a nice okay absorbance. pH 7 and 9, like red and green, they are good, out of which pH 9 is like the best. It is giving me the best OD reading. But pH 5 is causing some trouble. What is that? pH 5 is causing almost a deterioration of my AGNPs. And how do I understand that? The peak over here is becoming a plateau over here. So, a deterioration of the peak helps me understand that my AGNPs have deteriorated, they have probably disintegrated, the number of AGNPs has reduced, and this thing, the formation of a second peak over here from 420 to around 498, this is called a red shift. A red shift means aggregation of AGNPs, and this helps you understand which pH is useful for the formation of silver nanoparticles. So, like this figure is clearly telling me that pH 5 is just not to be considered. If you wish to consider, we should consider 7 or 9, not even 11, because 11 is again reducing my yield. Now, again, the same thing over here. Key dispersed nanoparticles give me a nice sharp peak, whereas aggregated ones of the same sample will give me a red shift. Here I am having it at around 420, and here I am having it at around 690, 650 or something, which clearly shows an aggregation of the nanoparticles. So the funda of this UV spec goes that the sharper the peak, the more are the silver nanoparticles, and the more the peak is at around 420, the more the tentative size of the silver nanoparticles will be around 20 to 50 nanometer max. But the more it starts going towards a higher wavelength, like 500 or 600, the bigger the size of the nanoparticle is becoming. Like it's aggregating such that it's no more a nanoparticle only, it has become a microparticle now, more than 100 nanometer, that way. And then this one is also telling the same thing, where you can see that AGNPs at room temperature, then AGNPs on heating clearly have an advantage. And here is where you see just the plant extract, where this is sometimes necessary to understand that when you are making biologically synthesized AGNPs, this is required. Why? Because sometimes people think that the peak is also because of the plant extract or the reagent that you are using. But no, you usually run the sample also, like the blank samples also, just to show that the actual extract or the actual reagent does not have a peak at 420, and it's only the AGNPs which are showing that peak. So yes, this is also required at times. Coming to dynamic light scattering. Dynamic light scattering measures the hydrodynamic size of the AGNPs. Now, what is hydrodynamic size? The AGNPs will have a certain particle size, like some size of the particle. And the hydrodynamic, this AGNP will be in a certain solution. Suppose it is in water, or suppose it is in ethanol, or suppose it is in some other useful solvent. Suppose NaOH, or suppose HCl, or whatever you like. So when it is in solution, it will interact with the particles of the solution, of the solvent, and it will form some certain interactions with the solvent. Now, the AGNP and the capping of the AGNP. The nature of the capping of AGNP will determine how it is interacting with which part of the molecule of a solvent. Like suppose if this cap here is negatively charged. These thread like stages are negatively charged. So they will interact with positively charged moieties of your solvent, and vice versa. Like if these threads on the AgNP are like positively charged, so they will interact with the negatively charged moieties of your solvent. So this thing will form a hydration sphere. Why is it a hydration sphere? Because they will attract certain molecules of the solvent towards themselves, and they will lead to a little bit of matlab, a little condensed sphere. Just not a proper sphere, but yes, they, here you will find a little more concentration of solvents than in the rest of the solution. So this thing will be called a hydration sphere, and the red thing that you are seeing is the actual particle, the actual AgNP particle that you can see. So ye particle plus that hydration sphere, this complete blue thing is then called the hydrodynamic size of the AgNP, where this R that you can see from the center of the AgNP, of the core of the AgNP to the end of the hydration sphere, is called the hydrodynamic diameter of the nanoparticle. So, this is why you want to measure the DLS. What does DLS tell? DLS tells you the uh, behavior of the AgNP in a solution. Now, it is possible that they can have a small particle size but a very large hydration sphere, or a very large TEM but a very, matlab, just a small layer of the hydration sphere. So, this tells us how the AgNP will behave when it is put in different solvents or solutions. It is also used to measure the polydispersity index. A broad peak means it is polydispersed, and a narrow peak means it is monodispersed. We'll just see what is this. So this is the DLS machine, where this is your sample. This is a laser diode, and the angle, the measurement is done in two angles. One is the 0 degree angle, like the straight one, how it happens in UV vis. The other angle is the 90-degree angle, where at 90 degrees also there is a detector which is sitting to detect any um rays or any wavelength coming from the particle. So this forms certain graphs, which is then finally using some Stokes Einstein equation, which is an equation to like which just simplifies these graphs into this simple graph to understand, where you just measure the particle diameter and the intensity. And here what it is telling, that a small particle, like similar to the UV vis range, that a smaller particle will have a, um, a dispersion towards the smaller this of value of the x-axis, and a coarse or a larger particle will have a peak at the larger side of the x-axis. Then the peak, like the area under the peak gives you a rough idea of the intensity of the AgNP, meaning a larger peak, a sharper peak means more number of nanoparticles formed of that particular size. Similarly, if the intensity is lesser, or if the peak or the hill is lesser, means the nanoparticles of this population are in a lesser number. So yes, usually what people aspire for is that this matlab aisa peak आना चाहिए in their DLS graph. This is what people aspire for. But usually what people get is they get a mixed population, like as you can see over here. So this thing, the presence of two different peaks, means this polydisperse. They are broad, so they are polydisperse. The presence of just this one peak, the blue one, would have indicated a narrow peak, so it would be monodisperse. And this is how an actual DLS graphs looks like. So see, you get a small peak, you get a large peak. So this large peak means more nanoparticles of this size range, less nanoparticles of this range, and the nanoparticle size range over here will be between 10 to 100 nanometers, and mostly it will be like around 50 nanometers or something. And the intensity is very high, means the area under the peak and the amount of AGNPs formed is very nice and very high. And then this is also telling like how different conditions can affect the particle size, like different pH conditions and something. Then zeta potential studies. What do zeta potential studies do? So we had seen this nanoparticle with AGNP will be a negatively charged Ag plus and I mean Ag0 nanoparticles. Suppose they will be covered with a negatively charged stabilizing agent. So this negatively charged particle will be covered by positively charged ions of the solvent, and this layer will be called something called a stone layer. Now this what will this again becomes a sphere of its own, and then because of this what happens, some more ions start to attract towards this bigger wala sphere, the one jisko we are calling stone layer, they start getting attracted to that, and then this becomes even bigger sphere than the ये stone layer से भी बड़ा sphere this one becomes. So the this wala plane, the biggest wale circle ka plane is called the slipping plane, and the interaction between these two spheres is called the zeta potential. The difference between the potential of this stern layer wala circle and the slipping plane wala circle is called the zeta potential. So as we can see it, as we saw in the diagram, it can be affected by both the AGNP also and the property of the solvent also. And it measures the stability of AGNP when it is dispersed in solution. It can be positive or negative. A higher value means a higher charge, which means a higher repulsion between molecules. This means that the AGNPs will not aggregate and stay as colloids. Meaning, suppose like this is a zeta potential graph of this, where they have measured this exact zeta potential between these two and they have portrayed it as a graph. Now, what is this graph telling? That this zeta potential over here in the red color, it is minus 31.7 millivolts. So, what does this mean? The result quality is good. What does this mean? That this zeta potential between this and this is minus 31, means the amount of repulsion between these two will be minus 31 millivolts. So the higher the value of the zeta potential, means the higher will be the repulsion between these ions and molecules. And because they repel each other, what will happen is that they will not combine with each other and they will not tend to aggregate with each other. So a higher zeta potential value indirectly means a higher potential stability of the AGNP and lesser chances of aggregation of the AGNPs. I hope this was clear. Then transmission electron microscopy. This is for measuring the size and shape of the nanoparticles. And yes, because we are measuring size, it gives you a size distribution as well. So TEM, as you guys must have probably studied in your BSc syllabus, it starts with an electron source. The electron source will give off electron rays, which will be condensed using these condenser lens. They will pass through this aperture, they will hit the sample. The sample will be hit and it will transmit and not reflect or repel. It will directly transmit the electrons through these objective lens, through the objective aperture into the screen. An adjustment of all of these, like the aperture over here, the objective lens over here, and the projected lens over here, all result in the successful formation of such images from TEM. But yes, the sample is here. The electron rays hit the sample, and after hitting, they transmit through the sample. So the sample has to be conductive of electricity. If it is not conductive of electricity, you will not get a TEM image. And this is a reason why the TEM images of organic compounds are usually not very clear because of this reason, because they do not have that much electrical conductivity. So this thing is used to measure the sample size of AGNPs. The scale is used to compare the sizes using
और कितने
software.
स्लाइड है मैडम?
और कितने स्लाइड हैं सर? बस 9 स्लाइड, 9, 5, 9।
प्रेजेंटेशन बंद कर दिया आपने?
हाँ,
शेयर जो
सॉरी
कर रहे थे
सॉरी।
वो बंद हो गया?
ओके ओके, 1 मिनट सर, आई एम सो
कर दीजिए,
सॉरी।
क्लिक कर दीजिए। ये दूसरा लेक्चर के लिए है?
अच्छा अच्छा।
नहीं, मैं बोल रहा हूँ कि दूसरा लेक्चर लेंगे क्या?
दूसरा लेक्चर?
हाँ
नहीं
मतलब,
सर, आई थिंक 8:30 तक, 8:40 तक मैक्सिमम हो जाना चाहिए।
ठीक है, ठीक है, कर दीजिए।
ओके। हाँ, सो दिस थिंग, huh, this machine can also be used for SAED analysis. So here this selected area aperture that you are seeing, this aperture is also used for SAED analysis that we will see later. So the TEM images, transmission electron microscopy images, what do they show? They show the exact size and shape of the AGNPs that have formed. The scale over here that you see, 50 nanometer. So you use this scale to measure the AGNPs over here. Like इसको measure करो, then इसको measure करो. So you will understand what is the size of the AGNPs. Then the shape also you can see, like here you can see it's roughly spherical, here it's almost cuboidal, here it's kind of hexagonal, here they are almost triangular or spherical. And this thing over here is called the size distribution, where different sizes are mentioned here and the number of particles of that size in this image are reported. So this is called a size distribution of the AGNPs. Now, SAED is done on the same TEM instrument that we saw before. It is seen as diffraction spots. The rings that are formed are similar to XRD patterns. You guys might have studied XRD in your syllabus. It differentiates between crystalline and amorphous phases. So, what happens over here is that this selected area aperture in this sample, when the electrons are transmitted through this selected area, if you fine tune this selected area, you will get these patterns, and each concentric, they're all concentric rings, and each ring is indicative of a certain phase. So what happens is that crystals are ordered structures. We saw already that silver is a phase cubic centered crystal. So they are ordered structures and they tend to have a repetitive pattern. Like, honeybees ka jo hexagonal shape is like everywhere uh, on their nets. So you see that everywhere, it's like a repetitive pattern. It helps you predict their properties. So the more the crystalline nature, the better you will be able to predict their properties. And that is why a crystalline AGNP is very much desired. Lastly, FTIR spectroscopy is to detect the reducing and capping agent for biologically synthesized AGNPs, obviously. And you need to run the actual extract which you used to form the AGNPs. And you compare AGNPs ka spectrum, you compare the extract ka spectrum, then a reduction in a peak of the extract ka spectrum means that that compound in the extract has been used for the reduction reaction, whereas a shift in the peak in the AGNP spectrum compared to the extract ka spectrum means that it has been used for capping or stabilization. Now, this is just the instrumentation of the FTIR, but this is the actual graph. So, what does this tell? So, red is for the AGNPs and black is for the extract. So, see, this peak got reduced, means this peak was used for the reduction of AGNPs. Whereas here you can see there is some slight shifting in the peaks. So these peaks were used for capping. Like even here you can see some very slight shift between the peaks. So these peaks have been used. The peaks, I mean, all these peaks correspond to certain functional groups. So we can say that these functional groups have helped in the capping of the AGNPs, whereas these peaks, these functional groups have helped in the reduction of the silver 2 AGNPs. So this was about the silver nanoparticle synthesis and characterization. I just want to show you that this was a paper that we had published in Colloids and Surfaces, in which I had reported my work on silver nanoparticle antibiotic combinations as promising antibacterial and antibiofilm candidates against MAR escape microbes. And the synthesis method of these silver nanoparticles had been patented, and the patent had been granted by the patent office in 2022. Thank you so much.